Carbohydrates sit and coat the surface of all bacterial organisms. They form a rich and diverse landscape of building blocks that go well beyond the standard carbohydrates that we see from day to day. There are different types of bacteria that might have, they might be even gram-negative with particular macromolecules on the surface. Because of the complexity, carbohydrates of glycobiologists have taken to representing every sugar by what I call a carbohydrate widget, saying nothing about its chemistry, but the slide would not be big enough if we were to try to show the molecules. But there are many, many different carbohydrates attached to lipids, attached to proteins, attached to large molecular structures, and even glycopolymers. And there's quite a diversity between gram-negative and gram-positive bacteria due to the cell wall structure, and there's a little bit of information about that. But what is most important about these structures is that this is the face of bacteria outside to the outside world. And all of these complex structures participate in communication between bacteria and other bacteria, amongst bacterial communities. You hear a lot about the microbiome. These teams all gather together in biological communities, and also between bacteria and the hosts that they infect. And in fact, indeed, they're very important with respect to one key issue. They are essential for bacterial survival, certainly laying down the rigid cell walls that protect bacteria from osmotic shock, but also they encompass many different virulence and pathogenesis macromolecules that, if intercepted, could potentially alter the interactions between bacteria and their hosts. So these are targets that are very interesting from a fundamental perspective to understand that communication between bacteria and cells and organisms, but also potentially have ideas about intervention into those pathways that are responsible for bacterial virulence and pathogenesis. You will see some of the time, especially in the beginning of this talk, a mention of a bacterial glycoprotein, an N-linked glycoprotein, because this actually formed my first entry into the world of bacterial carbohydrates, and it was a pathway that I'd been familiar with from the eukaryotic or human side, but now I started to see a simplified version of this pathway in bacteria, which really encouraged me to dig into the bacterial system. And I believe that every day I've worked on this system, I've learned new and interesting things. So when I read about carbohydrate complexity, I tell the people in mammalian biology, you haven't seen anything. 10 sugars kind of account for the diversity of sugars in human cells, on human cells, in the glycocalyx of human cells. But microbial diversity is exploded relative to eukaryotic glycan diversity because of these highly elaborated building blocks that are in many cases derived from simple sugars that we feature, like an acetylglucosamine, glucose mannose, but they are highly, highly modified. And the bacterial glycome encompasses, this is about a few years ago, this number's probably bigger, came from a database dive I did into a web server put together by Peter Tuchach in Russia, where he codified all of these sugars from pentose, hexose, all the way around to nonulose, and there are even some 10-carbon sugars. And also you see many different versions, furan, pyran, linear versions of the carbohydrates, both in the pentoses and hexoses. So this is huge diversity. This is just the building block. It doesn't say anything about how much diversity there is once you bring this large variety of carbohydrates together. You will see a little bit through the talk about this sugar called UDP vacilosamine, or di-N-acetamidovacilosamine, so I just point that one out. It's made from an acetylglucosamine. And then what I want to show you here is just a taste of a complex glycoconjugate, in this case the lipopolysaccharide with the O antigen repeat, where you see those usual sugars come together in even more elaborate structures. And these are a feature, an O antigen, which is often a calling card on a bacterial surface because the O antigen repeats are way out on the surface of bacteria. So bacteria use this glycoconjugate complexity as a rich language for communication. Can we understand it? Hardly, but can we dig into it as chemists and try to understand the logic of it? And that's what I really want to feature today. When you see the widget pictures, don't get confused that it doesn't represent something important. So this, for example, is the heptasaccharide that's featured on the surface of Campylobacter jejuni, and here is the one bacillocyanine, unusual sugar, that features within that glycan. We are fortunate to study bacteria because in bacteria, the genomic material is very frequently organized into operons. These are clusters of genes that may perform tasks within single pathways. So we can often look at those operons and say, wait a minute, I can see features in this operon, and I can start to dissect the function of the gene products in that operon. And that's very relevant within the protein glycosylation pathway of Campylobacter jejuni, where all these letters designate genes, and then those letters are also in turn designate the names of proteins. If in Campylobacter jejuni you disrupt any of these genes, you start to encounter changes within the bacterial physiology. So on this is simply a gel which shows proteins, so the dark bands are proteins from different molecular weights. But as soon as you knock out any single component in the pathway, you start to see the absence of those glycoproteins. This is the wild type or native Campylobacter jejuni. This is another protein that does not perform a critical path. And also, you can see in the physiology of the bacteria, if you knock out some of those genes, this is an in vivo activity assay for infectivity. You see an eradication of the colonization of the bacteria, in this case in a chicken cell model. So what I want to point out is that these pathways are essential for virulence and pathogenesis, and therefore they are intriguing targets for analysis. Oops, sorry. Okay, so let's move into the pathway. And I want to describe to you this pathway as a conveyor belt. It is an array of enzymes that work sequentially in order to build up that complex glycoconjugate, for example, the 7 sugars that are in Campylobacter jejuni. And I like to think of them as a polymer, from a polymer perspective, where these pathways involve certain types of steps where you can sort of categorize them as the way you think of initiation of polymerization and termination. So there is a priming or initiation step catalyzed by a phosphoglycosyl transferase, where that enzyme moves a phosphosugar to a polyprenol phosphate. I'm going to say a little more about this beast in a moment. Then there are elongation steps, where a number of enzymes string together additional sugars. Sometimes those sugars are modified, sometimes they're readily available. And then there's more or less a termination step, where that molecule is transferred as a unit to a lipid, to a protein, or to another segment of glycan. In all cases, these processes involve assembly on a polyprenol phosphate. I show you here the structure. I saw the odd isoprene in people's molecules through the last day and a half. Well, this is just a lot of isoprenes assembled in a linear polymer, and they vary in both cis and trans geometry, E and Z geometry, but they are absolutely unique to these pathways. So the assembly, this appears to be essential. Partly because of this, but partly because of the enzymes involved. They are membrane associated. For the average protein chemist, the minute you say my protein is membrane associated, is I don't want to work with you because they're too difficult. Membrane associated proteins pose additional challenges over soluble proteins. All of the assembly takes place on the cytoplasmic face of the membrane, that is inside the cell, because that is where all the materials are. but the later presentation of the sugar is on the surface of the cell. So it's no surprise that the assembly occurs on the cytoplasmic face, where the sugar donors are, the building blocks, everything you need. But it's really essential that a transporter or translocator flips that sugar structure over to the other surface into face the periplasm or the outside of the cell for transfer to sugar, lipid or sugar acceptor. And it's really no surprise that we have to work at the membrane, because you need to stick to the membrane, build up the sugar, leave it intact, somehow with assistance of a flipase and this, keep the sugar attached to that for its final transfer. So these are the prime characteristics of this pathway. Now, polyprenol phosphates. These are essential through all domains of life. They vary a little bit. They may vary in the saturation of this terminal unit. It may be saturated in the S configuration or unsaturated. The fully unsaturated appear in bacteria. But then as you think through evolution, they get longer and longer. So if you thought this 11 isoprene unit polyprenol was long, think for a moment towards plants and mammals, flies. They have even longer polyprenol phosphates. And in all those cases, that alpha saturation exists. So the variables are olefin geometry, stereochemistry or saturation of this terminal unit, and the overall length. And the one thing that is important to observe is that there is a variation of the geometry of the double bonds. They are not strictly linear polyprenols in an all-trans site. They're actually a mixture where there's a strong segment of Z olefins, and it's thought to adopt more of a helical configuration, which actually fluidizes the membrane. And there's a little bit of sort of thought on that. We studied the pathway with all trans but long isoprenes, and the enzymes just don't work. They really need that in the membrane. So here's the N-link pathway from Campylobacter jejuni. This will now recollect some of the things I've told you. In the early steps, there is conversion of N-acetylglucosamine to bacillosamine by 3 enzymes, an oxidoreductase, a transaminase, and an acetyltransferase to create this unusual sugar. That then is transferred, the sugar phosphate is transferred to the polyprenol phosphate at the membrane interface by this enzyme. It's a phosphoglycosyl transferase. Then as one walks one's way down the pathway, there are a series of enzymes that add sugar, 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 and they are usually of 1 or 2 folds and flavors, but they are each designated to do a specific task in a specific order. That finally gets you to the flipase to move that glycan into the periplasm in this case. Then that glycan is transferred in a block to a protein in this pathway, and the polyprenol diphosphate that is made is conserved and recycled. It's very energy efficient. It's recycled to end up back at the beginning with a pyrophosphatase. So much work has been done on all of the steps in the pathway. We played roles in assigning a lot of the biochemistry. But what's really interesting, despite the fact that some of these behemoths have been structurally characterized, most of these enzymes can be readily predicted through AlphaFold. They're simple folds, they're easy to predict. There's one enzyme that was always missing in action, that is the priming step, the first step, the smallest enzyme. And most of what I talk to you about today is going to involve that particular enzyme. And you may think, small enzyme for a whole talk? Trust me, it's intriguing. Okay, so I told you it catalyzes commitment to membrane. Another thing that I haven't indicated, but it's a reversible step. So it's isoenergetic, this pyrophosphate and this pyrophosphate, unlike the glycosyl transferases that are not. They're pretty much irreversible. This becomes important as one thinks about deploying cellular reserves of this important molecule. So it's the commitment to membrane. It has a unique biochemical activity. All the other glycosyl transferases just make glycosidic bonds. They may make them alpha or beta anomeric configuration. They may join different hydroxyls, but this one is different because it transfers a phospho-sugar intermediate. And it indeed is the signature of pathways that involve glycan assembly. If you see a phosphoglycosyltransferase in an operon, those collections of genes, then you say, okay, we're making a glycan and we're going to deploy those enzymes. There are in fact two structural superfamilies of these enzymes that catalyze the same fundamental reaction. What I will do is explain to you why I believe nature has evolved two superfamilies of enzymes. But this at the start is critical to mention. The most well-known of the phosphoglycosyltransferases are what we would call polytopic. They span the membrane many times. They bind the substrate on the cytoplasmic face of the membrane. You can see the sticks here of bound inhibitors. And the community has learned a tremendous amount about these enzymes through the availability of beautiful natural products such as muridomycin or tunicamycin, which are very strong by-substrate analog inhibitors of the polytopic phosphoglycosyltransferase superfamily. And these are well characterized, well appreciated. So they're often designated like this, where every block represents a transmembrane helix, and the action really is that the interface is between each of the helices, where those loops come together to bring in catalytic functionality and a binding site for the substrates. However, for the first time we started to realize that the PGLC that I just described to you belongs to a second superfamily that shares a common functional core, but is nothing like the other enzymes that are the polytopic. And this is what we knew about it. There are several members, but the smallest functional core, this little thing that does the same chemistry as these behemoths, actually is represented in a 200 amino acid residue protein with a transmembrane, purported transmembrane domain. And that was predicted through hydropathy analysis that said this looks like it spans a membrane, this looks globular. I mean, that's not a lot of information, but that was what we knew. This catalytic unit is represented in other superfamily members that may have additional auxiliary domains. To just reinforce to you before I move forward the importance of these phosphoglycosyltransferases, I want to call your attention to the diverse but all pathogenic, apart from, well, this may be pathogenic to chickens, but in general, they're pathogenic to humans, and many of them represent some of the escape pathogens, where there's a lot of drug resistance for those. They are in gram-negative or gram-positive and mycobacteria. Several of these are the escape pathogens, like Acinetobacter, going down, Neisseria, Salmonella, strep pneumonia. Many of these are serious human health threats. Another one of these on this list is Fusobacterium nucleatum, which is now known to be what's called an oncomicrobe, where it actually triggers oncogenesis. So in the polytopic phosphoglycosyltransferases, it's pretty clear, based on a great deal of information from those natural products, that the mechanism involves attack of the polyprenol phosphate on the beta phosphate of the nucleotide, and it goes through the intermediacy of a ternary complex. All that means is both substrates bind at once to the enzyme, and it's nothing fancy about that. And the structures of those proteins with different nucleoside analog inhibitors really speaks out to that mechanism being validated. But there are other metrics for validation where you see beautiful binding of these natural products to a key magnesium and acidic residues. So it's actually one of the key hallmarks of those enzymes. So here you see it a little better in more of a sort of complex picture of where the parts of the enzyme are organized. The enzyme inhibition is potent with very low micromolar, sometimes picomolar IC50s. Now to think about the mechanism of our small 200 residue protein, we thought, well, there's such a large difference in the size and topology of those proteins. Is the phosphoglycosyl transferase from Campylobacter jejuni just catalyze the same reaction in the same way? We had some doubts, but we had no evidence. However, while we were going through many, many attempts to solve the structure of that small protein, we realized through mechanistic studies that it was actually quite distinct with respect to mechanism. And that fascinated me. This is something one could really put some tools in to establish the mechanistic course beyond the phosphoglycosyl transferases. And what I will show you is the evidence for an aspartic acid in the enzyme active site attacking the UDP sugar to form an aspartyl enzyme phosphosugar intermediate, a covalent intermediate, which is then intercepted by the polyprenol phosphate to release the product. There are different hallmarks to this mechanism, but it's completely distinct, and the enzyme is pretty much being used in a very different way from the polytopic phosphoglycosyl transferases. And first of all, these enzymes are not well inhibited by bisubstrate analogs, like I showed you for the polytopic. Then I'm going to briefly describe these three types of experiments, which you can use to characterize the mechanism without a doubt. Mechanisms you have to be careful with, but I think we have this one pretty solidly locked up. And I want you to notice a couple of things. On the first step, you kick out uridine monophosphate to make the intermediate. But it's not till the second step that you invoke the polyprenol phosphate. So the first piece of evidence is measuring the production of uridine monophosphate in the absence of polyprenol phosphate. Do we see the first half? Is there some evidence? And we can actually quantify the release of UMP through an assay that characterizes UMP by luminescence, and we can show in the absence of enzyme, there's no UMP released. In the presence of enzyme, UMP released is robust and quantifiable. We could also show that if you use the non-cognate substrate, the substrate that's not meant to bind very well to that enzyme because they're specific for different flavors of sugars, what we see there is a much more attenuated release of uridine monophosphate, affirming that we have this first step that occurs without polyprenol phosphate. A second method, because you could just throw a rock at those experiments and say you're just hydrolyzing the UDP sugar to release UMP non-catalytically. The other thing that you can do that's a little bit more substantive is to do exchange of radio-labeled material back into the UDP sugar starting material. So if we dose in C14 labeled uridine monophosphate and follow over time, we produce radio-labeled UDP dianesopyl-glycosamine, proving that reverse step occurs in the absence of the UMP. And then finally, the most important diagnostic is to trap that intermediate. You can look at it and know that it's going to be pretty labile, so you have to trap it chemically, and that's usually done for a compound such as this, is done quite well by quenching in sodium borohydride treatment to capture this in the unfolded protein and then reduce the product. And what you get out of where an aspartic acid was, you get homoserine. So that's diagnostic of having formed that intermediate, and we can see that in the mass spectral MSMS analysis, short peptides, where you would have had an aspartate, there's a lower molecular weight corresponding to the homoserine. Okay, so those are our key pieces of evidence. We have another place where dichotomy of those enzymes, the difference in topologies, can come clear. And this is through work we've carried out in collaboration with Charles McKenna at USC, where his group has been making phosphonate derivatives of nucleotides to see how these non-hydrolyzable analogs behave with proteins. They're very good for structural analysis because you can trap complexes, but in the case of some of the phosphonucleotides that he has made, they've been actually deployed into therapeutics. So what the McKenna group did was they generated a series of bisphosphonates where both X and Y were hydrogen. They were hydrogen and fluorine with 2 isomers, and then there was actually, I don't know why I left it off, the difluoroanalog. And what I don't mean to read all this, but I think the most crucial thing is when compared with oxygen, these are not bad surrogates. Yeah, they're pumped up a little bit, but they haven't had a dramatic change with respect to sterics. And you can see that through bond lengths, bond angles, and distances throughout the complex. But what we find when we challenge the phosphoglycosyl transferases that we study is that the different bisphosphonates act differently with the 2 classes of enzymes, the 2 superfamilies of enzymes. So for the polytopic PGTs, and I speak specifically about an enzyme known as WEC-A, the bisphosphonate that's the CH2 derivative is the best of the lot with respect to binding and consequently inhibition. And the other bridge structures, both the R and the S monoprodeo monofluoro bridges, are about the same, and they're considerably, and then the CF2 bridging position is really much worse. These have an IC50 of about 41 micromolar, and really, I think the strongest evidence for the function there is that the sequential addition of the electron withdrawing groups and the increased basicity just improves inhibition to the magnesium that's at the enzyme active site. And so there's increased basicity improves inhibition. In contrast, and we were pretty surprised, I don't know what we predicted, the complete reverse order is seen for the phosphoglycosyl transferases of that monotopic type. And what we see there is absolutely non-existent inhibition by the methylene. Interesting differences between the R and S isomer at the bridging position, and the best bridging is CF2. And we now see that this is the strong effect in chirality. The IC50 is 32 micromolar, but I do want to point out that the analogues that were made, this enzyme uses UDP dianeacetamido-bacillosamine, but we only did the synthesis with glignac because to create a sufficient amount of that unusual sugar to walk through all of the chemistry means that it was pretty prohibitive at the time. So we just thought, well, let's just see what happens with the N-acetylglucosamine here. So I call your attention to the fact that in bacillosamine, this is just the 6-deoxy, and in bacillosamine, it's the 4' acetamido group. So this is another place where mechanisms are different and affirming the difference. And the other key point is it's only the alpha anomer that's active, the beta anomers are not. So there's sugar specificity and there's a divergence in the behavior of these compounds with the 2 flavors of phosphoglycosyl transferases. And so the studies reinforce that dichotomy. Here I want to just show you, now looking back again at the complex, you could see how potentially the methylene bridge here might have an impact on association with magnesium. Okay, so all this time, moving the mechanism forward, we still were trying to solve the structure of this rotten little protein. I mean, we grew gallons of protein and so on. And so we did some work comparing sequences and covariance analysis to at least say, can we develop a structural model? And to do that, what you do is you align many, many, many sequences of enzymes from a similar species, and then you look for similarities and dissimilarities. We could see that in all cases, there was a hydrophobic patch. There was this suggested transmembrane helix. And then there were other hallmarks of activity, which we did a covariance analysis of. Covariance simply means if you have a long polymer, protein polymer, and you change one residue, let's say this is an acidic residue that normally interacts with a basic residue. If you change one of them to a different charge, there would be a covariance where they would swap roles, so that you don't leave the dichotomy, you don't leave two positives or two negatives, the other one changes as well through evolution. And that's basically how covariance works. And we got a pretty crummy sort of globular model that showed no relatives in history. And the really irritating thing here is we had this bit, neither were very satisfactory. And what's even more unsatisfactory is you don't even know how they're associated with each other. And therefore, you have a big question mark on the structure. But I'm going to put you actually completely out of your misery, because finally, in 2018, we were able to solve the crystal structure of the phosphoglycosyl transferase from a related species of Campylobacter C. concisus. It's the crystal structure of the residues 1 to 183, so that's short of the 201 full length. There's actually really more of a terminus here that comes out. The structure nicely captured a phosphate ion and the magnesium. But this was carried out on detergent solubilized protein. So when you have a membrane protein and you want to do X-ray crystallography, you have to take it out of the membrane with detergent and crystallize those detergent extracts. We know that's probably not good. It's going to be disruptive if you're peeling away native lipids, but nevertheless, if that's all you have. If this had been a far bigger protein, we could have tackled with cryo-EM approaches, which we're doing now, in a membrane environment. But this was the small protein X-ray crystallography. There were there were features of the structure. There was this, what we call now a reentrant membrane helix, which dips about 40 angstroms into the membrane, and it has a highly conserved kink in that helix, which means the helix does not go transmembrane, but dips back out again. So that's a novel finding, although some reentrant helices have been characterized by NMR analysis. They've not been seen in X-ray analysis. Another important feature is where the magnesium and the phosphate is exactly where the catalytically essential aspartate glutamate diad is. And the two caveats are, we're missing a tail. Now, losing 18 residues isn't too bad if your protein has 400 of them, but losing 18 residues when it has 200 feels a little bad. It's like a 10% loss of information. And then the other thing was that this structure was in a non-native dimer structure. And by that I mean, when the protein is in detergent, it forms a dimer where the loop that I show you to your left intercalates into the dip in another monomer. So it's a little bit irritating because people are going, well, that doesn't look right. I mean, how can you claim this is a reentrant membrane helix? How can you claim this structure? It's not something we've seen before. So we went to enormous lengths to establish the validity of that reentrant helix. In fact, at that same meeting that Sonsoles showed you a picture of, we talked with her and had her help us with MD simulations in membrane. And then we did a lot of other biochemical transformations and experiments to prove that topology. So what you see here is the enzyme has its feet in the membrane, a globular domain outside. It's a fairly open structure. And then when we looked back at covariance at a much deeper level than usually done, you can start to see interactions between helices that are not in the membrane with that reentrant membrane helix. There's a lot of auxiliary information that was all carried out using complementary biophysical analyses, which is really what you need to do for these types of challenges. Can't claim it till you can claim it. Now, I expect most of you have been hearing about the AlphaFold explosion, where people go, oh, I don't need to solve a structure, I'm just going to do AlphaFold. If we had put this structure into AlphaFold, without our structure being in the protein data bank, the structure would not have been predicted. So you have to have as many diverse structures as possible to make AlphaFold valid. Now our structure's in there. The good news is you can do something with AlphaFold to predict the structures of phosphoglycosyl transferases that use different sugars. And I'll show you that in a moment. But what was interested in AlphaFold is with the structure in the PDB, we went back to AlphaFold because we were still bothered by this 18 residue omission. And what we could see in AlphaFold along this axis are the positions within the protein 1 through 201. And this, the green suggests how well covered the sequence. Do you have tens or hundreds of analogues of the sequence that you can compare? And what we noticed is that there were a set of phosphoglycosyl transferases where sequence coverage was pretty good. And then I turn you to this map. This map tells you reliability of the fold that's predicted. And what you can see here, there's pieces of the fold that are really well predicted. They're right up at the highest amount here. Once again, residues 0 to 200. And then there's this big dip, which is that floppy loop. But we recover some order. And what was intriguing, if you then refine the crystallographic data, what you start to see is that you can bring that long missing strand in to a cluster that forms an aromatic box. And I'm not sure if people all know what they are, but they're really cool motifs. So a terminal phenylalanine in the sequence, phenylalanine 197, just 3 from the end, actually completes the aromatic box. Let me show you that. So that's where the aromatic box is. And then this would be the active site. Who knows what's going on here? If you change any of the aromatic residues for an aliphatic residue, the enzyme's dead, suggesting you need to bring together the 5 aromatic residues in the aromatic box to stitch this loop in place as you start to form the active site. And I'm going to show you just how much I like the aromatic box. Here it is in close-up. Those are those 5 residues. You see them all coming together. There are a lot of rules based on analyses that really show you what will make a stable aromatic box. And in many cases, there are perpendicular or parallel interactions amongst the aromatic groups. And we now know through a little more sequence gazing that this tyrosine and this tryptophan, which are sort of close to where sugar binding is, may well be involved in sugar binding. The other difference, if you look, we kept on finding new surprises within the structure. We even to the extent that people often thought that the phosphoglycosyl transferases that we study, which have an aspartate glutamate dyad, the polytopic having aspartate dyad, they said, no, they must be the same, they must do the same thing. But if you look at the structure, those 2 acidic residues are posed completely differently. In the polytopic, they are on a traditional alpha helix, but in PGLC, they're actually at a junction to a 310 helix, and those 2 acidic residues actually move together more primed for the catalytic steps, whereas if you're on an alpha helix, you're more apart. These may seem like subtleties, but what was exciting was that there was so much information in that one structure that we could begin. So we really have unlocked the superfamily with the aspartate glutamate dyad, the reentrant helix. We have this mobile loop I'll mention to you in a minute, and the aromatic box. Now, phosphoglycosyl transferases work on many different nucleotide diphosphosugars. We've mostly seen the uracil-based ones, but there's no doubt other nucleotides might operate. Additionally, and similarly, they use different sugars. So there are many, many monotopic phosphoglycosyl transferases that are going to diverge in the point in the structure where the sugar binds, okay? So here's an alignment of a lot of sequences of enzymes that bind to dianesitamidobasillosamine, and we see the residues that are critical for catalysis. They shouldn't change. One phosphoglycosyl transferase should have the same catalytic residues as another. But where they diverge is at the residues that are responsible for forming the sugar binding site. So for example, there's a tetrad glyLULU-PRO. It's abundant in the enzymes that use dianesitamidobasillosamine, non-existent in the very low in a family as a whole. And the other thing is that what's really cool is that that aromatic box is associated with the dianesitamido enzymes, utilizing enzymes with those 5 aromatic box residues. Now, if de novo you looked at this sequence, you would never predict that those residues are close in 3-dimensional space. But we now know that they are, and that's a great flag for a particular class of phosphoglycosyl transferases and their substrate utilization. And in fact, we were able to do quite a lot of work by aligning different phosphoglycosyl transferases that we either knew the sugar substrate or didn't know the sugar substrate. And we can see that the cluster that includes a lot of enzymes that use diNAC-back cluster in a very similar place in this dendrogram. So we have now generated hidden Markov models to start to distinguish which enzymes have which substrate specificity. And we were able to even get into the B subtilis pathway in collaboration with Nicola Stanley Wall to actually find the function of the first enzyme in the Bacillus subtilis biosynthesis pathway, which actually also uses bacillosome. But the truth is, looking at this clustering, what you really want is a much huger scale so you can start to pick out the specificity of all the others. Why is that important? It's important because it's the first step. If I can catalyze, if I know what the first step does, I can make the product of that step, and then I can look for what the next step does. So it's a real hallmark that you can use once we can dissect specificity of far more enzymes. Just to before moving over, I just want to mention an interesting thing about the PGLC. Unlike a lot of proteins that associate on one face of a membrane, they are usually enzymes that have evolved from a soluble precursor through evolution. And that enzyme has changed a bit to achieve a hydrophobic face, and then they park down on the membrane. This enzyme hasn't done that. It evolved independently because it would not have had a soluble precursor because the structure is so reliant on the reentrant membrane helix and that docking at the membrane interface. So what we now have is various disguises of the phosphoglycosyl transferase that have gone nicely beyond this nasty topology map. And what is really nice to see is by solving this structure of the smallest member, we can now look at that catalytic unit in the other members of the superfamily. And the specificity for sugars is all encoded within this unit. The other components are due to different things. They may perform different functions. For example, this is a regulatory domain, or this may help anchor to other proteins, but we can get much more clarity there. And the other thing that's important, and we dug into this, is that the catalytic efficiency of the two types of enzymes, polytopic versus monotopic, is quite different, and that is significant with respect to physiology. The catalytic efficiency for the small monotopic PGTs is about 75-fold higher than the catalytic efficiency of the polytopics, suggesting that these enzymes are much more efficient and can work at much lower undecaprenol phosphate concentrations, as suggested by their catalytic parameters, than the polytopic. So this tells us that these may have evolved for different functions, and one is more efficient, maybe operating under limited conditions. One is less efficient, but can then carry out many tasks easily. And so what we would put forward here is that the two topologies that we have now defined actually may be important for segregating functions of glycan biosynthesis pathways. The two topologies are found, maybe two polytopic, two or three monotopic within one bacterium, but this may represent a way to differentiate pathways. So let's say we have a polytopic PGT that's responsible for peptidoglycan biosynthesis. When there's an abundant, and the polyprenol phosphate is limiting, you may want to turn on the pathway to make peptidoglycan because you need it for new cells. Every new cell needs a batch of new peptidoglycan. But in other cases, the cell may be fully formed, but you want to display important entities on the surface. And in that case, the bacteria might choose to turn on a different pathway where you make these specialized glycoconjugates that are important for virulence and pathogenesis. That way, even though bacteria are not compartmentalized, they can do different jobs with two different topologies of enzymes that have different catalytic efficiencies. And this diversity may enable allocation of a supply of the undecaprenol phosphate to what are known as metabolons, clusters of enzymes that work in a pathway and associate with each other. And this would be an idea of a metabolon, and it really suggests that bacterial pathways, even though they are not compartmentalized, the enzymes may come together in larger groups to ensure efficiency of sequential steps to grow a glycan epitope or glycan polymer. What you don't want to do with a set of enzymes like this is have them all function independently, because in each case you would make a product. If it diffused away from the enzyme cluster laterally into the membrane, you'd then have to do a rebinding step, and it would reduce efficiency greatly. So it's keeping the conveyor belt going. So the interactions are probably important and limits intermediate release. There's importance of regulating the pathway, so you're very careful with the undecaprenol phosphate, and then there may even be post-translational modifications that may also regulate the pathway. So I've shown you a number of things so far, but one thing I do want to stress is that while the polytopic PGTs are bacterial and eukaryotic, and all your N-linked glycoproteins are made through a pathway that has the very same logic that the one I described, the monotopic ones are uniquely bacterial, which then does support the possibility that they may be very interesting targets for inhibitor development. So let me move into a sort of the almost last phase of what I want to tell you. So we realize that there is such a sparsity of discrete information on glycan structures in different organisms, despite the fact that those glycans may be essential for bacteria infecting human hosts, for example. And the traditional methods are very labor intensive. Often in a traditional analytical approach, you have to grow a culture, you have to purify out a glycan epitope, a chunk of glycan, then you have to do very serious two-dimensional and multi-dimensional NMR analysis to deduce the structure even of a 4 or 5 sugar unit. These are heroic efforts. Some have been characterized, but very, very few. The other problem with doing a genomic approach is that traditional genomic approaches, you may think that you know an order of enzymes, but there's actually misassignment because you didn't know what the first sugar in a repeating unit was. So what we posit is that an advantage is to go from the bacterial operon and actually look at the enzymes that describe the sugar or the glycan. You can pick out the enzymes that make bacillocimine, you can pick out all the glycosyl transferases, and now you can pick out the monotopic PGT with a surety, and PGLB is the final transferase. So the newer approaches that we're trying to do is to deploy bioinformatics and a biochemical pipeline to start to say, can we take an important bacterium of therapeutic, physiologic interest and read the glycan from the genes? Genomic information we have, it's practically coming out of the walls. It's protein and sugar information that we don't have. So what I'll show you is how we start to deploy this pipeline by, first of all, an integrated approach to define specificities of other phosphoglycosyl transferases, which open new opportunities for a cadre of different things. And the approach I'll describe to you is what is known as the formation of sequence similarity networks. This set of bioinformatic tools was developed by John Gert and Patsy Babbitt in the Enzyme Function Initiative. And this is a large consortium of people developing tools to allow you to assign not the function of one enzyme, but the function of large clusters of homologous enzymes. And the way a sequence similarity network works is if you have sufficient sequences, you can start to cluster them in pairwise across all of those sequences, not just one by one or looking at 1 and 10 others, but pairwise over tens of thousands of sequences. and you can set the stringency for how similar they are. You can set those values, these are designated as E-values, to see the stage at which you cluster out monofunctional clusters with the same substrate specificity. So this would be the workflow. We started in this case with 32,500 sequences. They were all the small monotopic PGTs. We did an E-value analysis to develop the network. At the bottom of this, you would have isofunctional clusters, same mechanism, common substrates, and then looking at other clusters, they would be same mechanism, different substrate. And the only limit here is the number of sequences that are non-redundant, and also your computer power, because if you have a very large network, it's going to start weighing down some of that. So Theo Durand in my group, who was just an undergrad from Imperial College for a year, took on this entire challenge, and he gathered together all the enzymes for the sequence similarity network, and every dot on this picture represents a sequence. And when the sequences cluster into lumps like this one or this one, then you feel that they're probably isofunctional. Theo did not cut corners, and he did what's called a closeness analysis, because he wanted to see how the clustering behaved as you varied that stringency. And he said, well, I don't think this value is good enough. I'm going to go higher stringency. And this was the day where we all gasped, because you can see beautiful clusters of isofunctional phosphoglycosyl transferases that would be predicted to use the same substrate. In some cases, they're a little lumpy, so you're not sure if the whole core is like that or just a core that is sort of more centric here. But every cluster with a number now has an assigned UDP sugar substrate. The bacillosabine cluster is actually this one, so it's a very abundant cluster. As you go down, you see all kinds of other strangers down here. Do we need to do more sequence similarity network? What we're doing is we're adding more sequences to see if we can start to embrace a few more isofunctional clusters. You can also look at these on a dendrogram, and I can just show you here some of the clusters we have assigned. Bacillosamine 1, then 2, glucose, 3, galactose, 4, down here, a keto sugar, which is both an intermediate and a sugar that is transferred. And as I said, every cluster with a number has been assigned a structure of its substrate specificity, but it's predicted. So not to be sloppy here, we thought it was essential to really define biochemical activity in vitro, and we used a new method which we call a CEF glow assay. This assay uses the measurement of UNP as the output for the enzymatic reaction, but rather than isolating all the enzymes, all the phosphoglycosyl transferases, and having to deal with purifying them and then being unstable, we assayed them in a cell envelope fraction that was its native place, so we could get high activity with, frankly, minimum work. And we were able to do kinetic analysis in this CEF fraction, and I can show you that it was very well validated for both stringency and validity. And through that, this is how we looked at all of representative members from different clusters, and were able to assign different sugars to the clusters. This is the library of sugars we're using right now. We'd be delighted to add more UDP sugars, possibly TDP sugars, which are harder to get. And these come from commercial, obviously the easy peasy ones are commercial, chemoenzymatic or synthetic, or a mixture of the two. So some of these can be more readily prepared by chemoenzymatic approaches like Dynacbac and Fucnac4N than they can by chemical methods. Although in the early days, when the stereochemistry of bacillosamine was not clear, we did that through chemical synthesis. So you can see we feel pretty secure about this assignment. Just one caveat about the glow assay. It's proven very reliable to give substrate specificity, the assignment of a particular reducing end sugar in a biopolymer repeat. But occasionally you might predict that an enzyme in a cluster uses a sugar, in this case WREU, from Rhizobium etli. We thought, okay, that's what it is in the glycopolymer. This must be the substrate. Nature pulls tricks on you, and it actually wasn't. The substrate for the phosphoglycosyl transferase was the keto precursor for quinac, and in fact that is what the PGT transfers, and then once it's attached to the polyprenol diphosphossugar, then it becomes quinac. So it's a step that happens later, and you've got to catch these possibilities. One way to catch them is through an analysis known as genome neighborhood network analysis, where you'll look at that genome, the operon, and say, is there something in here that might make an unusual sugar? And so, for example, I'm just showing you the PGL, the operon from Campylobacter jejuni. But here we can ask which genes are near this gene, and we can say how many genes away. But you see all the bacillosamine enzymes, PGLF, ED, the oxidoreductase, the transaminase, the acetyltransferase. So we know that substrate is available. And we can go back to the example I just showed you and prove that there was an enzyme that was capable of doing that oxidoreductase step to put the quinac in place. There was a membrane-associated enzyme. So this just shows you, looking at more operons, I can pick out any monotopic PGTs. And I can also find the polytopic PGTs. So these operons really write the glycads. We just have to decipher them. Can we go forward and do this with a bigger scope of enzymes, the glycosyl transferases? We're nibbling our way pretty well in to the next sets of glycosyl transferase GTB fold to do a similar thing, but it's on a much larger scale, and there's some trickiness to it, which I'd be happy to elaborate on, probably over a glass of cider or something. Okay, so in each of these pathways, you spot the PGT. That gives you a clue to the glycoconjugate, and when you're in this situation where you have a glycopolymer that's segments of repeat, you can actually now know, okay, this goes first, this second, this third, then there's a branch. So you can decipher things more at a distance once you know which the PGT step is. Okay, let's see where we're on. We have two classes of nucleoside analogues. I've mentioned the behavior of these compounds, which I think are wonderful mechanistic and structural probes. The other tool we have in our deck is neutral nucleoside analogues, which build off, in this case, uridine, but we've done them for some of the other nuclear bases, where we've established a solid phase synthesis of these compounds to create a lot of diversity in a nucleoside analog that can be screened against bacterial preparations or enzymes. The reason why the solid phase synthesis approach here is so important is nucleoside intermediates are a devil to purify. So we thought we could take advantage of all the advantages of solid phase synthesis by doing the assembly with common conjugation reactions like click chemistry, amide formation, and so on. So Christine Alber in my group developed a very nice synthesis. We made the amino, the 5' amino uridine, anchored it to a solid phase synthesis resin, where we could then have that in place for growth, for constructing new intermediates from this secondary amine. And through the solid phase synthesis, we can generate a lot of different analogues for testing against different phosphoglycosyl transferases, but any other enzymes that use nucleotides. We have a library of about 120 compounds, which we distribute to colleagues. They don't need much to do assays, so 10 microliters of 10 millimole will last them a lifetime. And so we can deploy that CEF glow assay now to see, can we screen for inhibitors with that assay? And indeed, when we put together a series of phosphoglycosyl transferases that we screen against different inhibitors, we see different inhibitor sensitivity and pick out an optimum structure for a particular flavor of phosphoglycosyl transferases. Then we can go back to that structure of the inhibitor, diversify it more, and keep optimizing the inhibitor. And through this approach, we have inhibitors that have IC50s in the 2 to 5 micromolar range. So we're moving quite well down the concentrations. The other thing that we can do with the structure that we have is now go to AlphaFold and predict the structures of other phosphoglycosyl transferases that use different structures. Now, I know looking at this is really annoying, and I'm not sure everyone can distinguish between magenta and green, so I prepared a more powerful way to look at this. All right, so here are going to be 5 phosphoglycosyl transferases that use different substrates. And in green here, that is the original Campylobacter concisus structure we solved. So the galactose one, you can see it's quite different. It's got an extra appendage here. The N-acetylfucosamine one differs again. The GalNAc one differs again. There's subtle changes there. I'll point them out to you more in a moment. And finally, the glucose one differs again, and let's disappear the glucose one. And you can now, by eye, with AlphaFold, say, oh yeah, I can tell this is galactose utilizes, it's got that huge helix turn helix. Or I can tell this one does something else because this helix is a bit longer. So we can now codify enzymes by even just structural analysis. One last thing that we've deployed is molecular dynamics, both to get an orientation within the placement in a membrane, but start to see the interaction of the phosphoglycosyl transferase with the polyprenol phosphate substrate, where the way we did this has been done in collaboration with John Stroud at BU. We can strip off the detergent from our model that came from the X-ray structure. Oops. Park it in a membrane, or strip off the detergent, and then park it in a slab of membrane that's the appropriate composition for the bacteria, and we can put in the undecaprenol phosphate, and then we can see the pose and do molecular dynamics to understand the binding of the polyprenol phosphate to the protein as a whole. And then we can do experimental validation. And what we see from that analysis is on this graph, you have the residues of the phosphoglycosyl transferase, the carbons of the undecaprenol phosphate, and you can actually see in the molecular dynamics run, we put in 2 molecules of undecaprenol phosphate, but one literally snuggled up to the phosphoglycosyl transferase, and you can tell that by these contacting residues that are all along the length of that polyprenol. You can also tell that this remains in one leaflet of the bilayer, which is actually an advantage, because disrupting 2 leaflets is more expensive than disrupting 1 leaflet that's already been disrupted. And we can do, from the molecular dynamics, we can do simulations to look at this loop and its movement, and we have also been able to compare it with some experimental data. And what we see is that the simulations nicely predict the experiment nicely predicts the simulations. I shouldn't say the other way around. And they have the same principal component, even though the movements in our experimental data, because it came from an X-ray analysis, were a little smaller. There's a clear motion of that loop, which has always bothered me, because what's that open active site doing? And I just want to show you that those motions can now be recapitulated for some of the other PGTs. In this case, the one that uses galactose, you see that helix turn helix. This is in membrane, it's being analyzed with substrates present. And then if you look at the substrate bound version, I want you to notice this is a triplicate run, that the enzyme closes up and then holds that reactive intermediate long enough for it to be intercepted by polyprenol phosphate. It's not an open active site. We never thought it would be. So in our evolving view of the structure now, we started off with an X-ray that was missing 14 residues. With the gracious help of AlphaFold and a lot of tricky modeling, we were able to now get that C-terminus piece in place. And now, through computation and experimentation, we can get the fully closed structure. What will that allow? It will allow us to determine all the substrate binding determinants, one by one, to say, how can we make our inhibitors better? That's what we need a structure for, because in the almost 10 years since we solved the first structure, we haven't solved another structure, despite enormous efforts. What we have solved is both the bifunctional and large monotopic PGTs, but by cryo-EM, which is a lower resolution output. So this was a long time coming, but this is the model of the closed structure from from substrate binding into the MD analysis. Now we'll move in. Here's the magnesium. Here's the pyrophosphate, glutamate, aspartate, active site residues. And this was the gift from the PGTs. There is wonderful uracil binding with the methionine on one face, phenylalanine on another, and the arginine guanidine just pinning it, in place. So that's a nice uracil sandwich. And in the novel, we can also start to see the carbohydrate binding now in that other part of the protein. And from this, I just want to point out to you, that is where the aromatic box would be. It's almost in reach of the sugar, and I think there's a good reason to believe that it's involved. This is probably not terribly accurate, but it's where we're thinking so far of the involvement of residues, with the residues that are involved in catalysis, a whole swath of arginine basic residues that are important to create a very positive area for a negative substrate to come into, especially at the head of the phospholipid head groups. And you can see then the phenylalanine, methionine, arginine, and now we see within reach tryptophan 118, which is in the aromatic box, and tryptophan 150, which might also serve to start to recognize the bacillose. And this is an electrostatic picture, just shows you that sort of wealth of basic residues that more or less catches and attracts in, steers in the negatively charged UDP sugar. Okay, I'm almost done. Let me tell you what I feel we have learned. First of all, there is a lot of genomic information and we need to exploit it. We can't go directly to carbohydrate information, but with informatics, you can start to put together the trajectory of pieces that make up the glycan that is made by those sets of genes. And if you have a particular organism that you're very interested in, it might be new or it might be disease related, you can certainly, with our tools right now, dig right in and say, this uses this sugar substrate, or we need to figure out which one it uses. The new challenge for us now is predicting glycan products from GTA and GTB fold transferases. Can we really read the operon to make the epitope? Glycan epitopes are pretty important. They number maybe only 3, 4, 5 carbohydrates. We don't need to make a 14 sugar unit. We need to make those small repeat units to define the glycan. And these would be useful for vaccine development, glycan arrays, and glycan binder evolution. So there's a lot of uses if we can now predict those set of sugars. And with respect to glycan biology, certainly inhibitors will advise understanding glycan biology. They may provide some therapeutic leads, but there'll certainly be probes. And I think that the inhibitor discovery using biochemical and phenotypic screening is a place that we will really dig into now that we're starting to improve the binding of our nucleoside analogs and make them better. And the one last thing, this is sort of out of sequence, we also want to convert that dreadful UBP synthesis into a solid phase synthesis, and I think we'll be able to do that there, because that will give us more access to that chemotype. This is the team, this is the blended team. I mentioned Karen Allen at the beginning. It's me, Christine, our famous peptide chemist, who did all solid phase. Shomi, who now has Christine's at Eli Lilly. Shomi, who now has an independent position at the University of Virginia. Some of these other characters are still here. Greg, Dodge, and Hugh know everything about AlphaFold and actually seem to already know when there's a new program coming out, because I think they stay up all night reading bioarchives and things like that. And then there are key players in both the Allen group and in my group who've been involved either in the past or now in this project. I'm done.